Hey everyone, welcome back. This is the screencast for the endosymbiotic theory and plastids. We're going to start today by going over two organelles that you may have learned about earlier in class, but we're going to learn about them in a little bit more detail. So first and foremost, make sure in order to prepare for this screencast that you have out your Unit 4 notes. You should be on page 11 of those Unit 4 notes. And you'll notice that we're going to actually start out at the bottom. We're going to define this term plastid, and then we're going to go through two examples of plastids, which are chloroplasts and mitochondria. And we'll wrap up today with a quick video about the endosymbiotic theory. As you watch that video, make sure that you take notes on the bottom part of page 11. All right, let's get started. So first and foremost, we'll learn that inside of a eukaryotic cell, so if you remember, eukaryotes are true nuclei, our organelles are cells that have true nuclei. We learned about all of these different organelles earlier in the year. And we learned that inside of both animal and plant cells, or autotrophs and heterotrophs, we have DNA. And we learned that DNA is stored in this one spot inside of the nucleus, here and here. What you might not be aware of, though, is that that's not the only DNA we have inside of our cells. We also have DNA in two different organelles within our cells. So we have DNA inside of this, our mitochondria, and DNA inside of our chloroplasts. In addition to that, we'll learn that those mitochondria and those chloroplasts are a little bit weird. They have some characteristics that are a little bit unique to all of our other organelles. And we'll learn about why those characteristics are significant in just a little bit. So let's start off by defining what a plastid is. So a plastid is any organelle that has a double membrane, has their own DNA, and is somehow involved with the creation or processing of food and energy. So if you think about it, that kind of describes two organelles that we've learned about so far. And those two organelles are listed on your page 11, the chloroplast and the mitochondria. So let's talk first about the chloroplast, and you can fill in the notes on the left-hand side of your page 11. The first is that chloroplasts are only found in autotrophs. And we learned that they have a pigment inside of them that is green, that gives the plant its green color. And we call that chlorophyll. The chloroplasts have an inner and an outer membrane. We learned this in class. That's super significant because the membranes look slightly different from each other. The inner membrane looks very much like a eukaryotic cell's organelle. The outer membrane, we're going to learn, looks a little bit different. Looks like more like a prokaryotic cell. Inside of the chloroplasts, we have these many little stacks of thylakoid called grana. And inside of those grana, they capture electromagnetic energy and convert it to chemical energy in the form of glucose. We believe that because of the weird characteristics of the chloroplast, that they might have formed due to this thing called endosymbiotic theory. And we'll talk about what that means in just a second. Lastly, the chlorophyll actually have their own DNA. So this is different than the DNA that you get from your parents, your biological parents. This is DNA that is completely different than human DNA. It looks closest to bacterial DNA, and that'll become important when we talk about how we think these chloroplasts came to be in plant cells. So let's skip over now to the right side of that column of your page 11 notes, and let's talk about the mitochondria. Now, the mitochondria are found in both autotrophic and heterotrophic cells. We discussed this and how it was important because despite if you're a plant or an animal, you still need to make ATP to move things within your cells. So we need the mitochondria to do that. It is the site of cell respiration, which is the reason why plants and animals both need them. It also has an inner and an outer membrane. You remember we talked about those folds in the inside part of the membrane are called cristae, and that's where those two parts of cell respiration take place. So the Krebs cycle and then the electron transport chain. Inside of the mitochondria, we convert glucose into ATP, and we will see that we have more mitochondria in areas where more energy is needed. So a great example of that is that we actually see more mitochondria inside of muscle cells than we do inside of like a skin cell. The reason for that is that muscle cells require a lot of ATP. Every time we move our body, we use muscles, and that is very expensive. We need to use a lot of energy in order to move those muscles. If you compare that to like a skin cells, sure, they act as a boundary. They protect us from the outside world, but they don't have to use as much energy. Another great example of that is that a lot of sperm cells are found, or a lot of mitochondria are found in sperm cells. And that's because sperm have to swim very, very quickly in order to fertilize the egg. So moral of the story, mitochondria, wherever a cell needs more energy. We also feel that the mitochondria has been formed because of this thing called the endosymbiotic theory. And mitochondria also have their own DNA. So let's talk about why that might be important. Why does it matter that both mitochondria and chloroplasts have two membranes? Why is it important that they both have their own unique types of DNA? And why is it important that they're both involved in energy? And the answer to that is this term endosymbiosis. So let's break that down like any good biologist. We'll break it down because biologists are not creative when we name things. We name things after exactly what they are. So this term endo means into. All right. You should remember that term from our unit 3 when we talked about cell transport and bulk transport. We move things into our cell. It's called endocytosis. So endo means into. Symbiosis sounds like the word symbiotic. And that's a term from 7th or 8th grade biology class where you learned that some organisms have what's called a symbiotic relationship. It means how they relate to one another. And if something is symbiotic, it means that both organisms benefit. So let's talk about these two words and how they can be smashed together into this idea of endosymbiosis. So here we have down in this image, we have a eukaryotic cell. How do we know it's eukaryotic? In the very center, there's our nucleus. We can also see that it has a membrane and a membrane around the nucleus. So we know this is a eukaryotic cell, meaning true nucleus. And right next to it, we have this aerobic prokaryote. All right, aerobic means that it can make energy not in the presence, in the presence of oxygen. And what that means is that this prokaryote, this little bacteria that's swimming next to this giant eukaryotic cell, is able to make more energy in a quicker way than our big eukaryotic cell. So what does the cell decide to do? It absorbs it. And you can see it enfold that membrane into a little pocket and it makes a vesicle around that bacteria. And what we notice there is that first we had one membrane around the prokaryotic cell here, but when we folded that inward, it made a double membrane. And that's where that second membrane comes from. This aerobic prokaryotic cell, guess what that turned into? That is now what we call our mitochondria, right? So this eukaryotic cell absorbed another prokaryotic cell, literally ate and swallowed a bacteria, and now we call that bacteria a mitochondria. So we think that that's where this organelle came from. Similarly, if we look at this is a photosynthetic bacteria cell, a photosynthetic prokaryote. And this eukaryotic cell said, it'd be pretty handy to be able to go out into the sunlight and make sugar on my own. So they also absorbed that prokaryote. Now you might say symbiosis means mutually benefit, meaning what did the prokaryote get from being swallowed by the eukaryote? Well, prokaryotes are tiny, which means that they gained a little bit of protection. They also gained a lot of exposure to nutrients because eukaryotic cells are much larger and they're more efficient at diffusing and transporting nutrients into their cells. So the prokaryotes gained a nice nutrient-rich place to live, and the eukaryotic cell gained the ability to make lots of ATPs from the mitochondria and make energy from the sun from the chloroplast. So let's learn a little bit more about this theory of endosymbiosis. We're going to watch this quick video clip together. Make sure you're taking notes at the bottom of page 11.
What if you could absorb another organism and take on its abilities? Imagine you swallowed a small bird and suddenly gained the ability to fly. Or if you engulfed a cobra and were then able to spit poisonous venom from your teeth. Throughout the history of life, specifically during the evolution of complex eukaryotic cells, things like this happened all the time. One organism absorbed another, and they united to become a new organism with the combined abilities of both. We think that around 2 billion years ago, the only living organisms on Earth were prokaryotes, single-celled organisms lacking membrane-bound organelles. Let's look closely at just 3 of them. One was a big, simple, blob-like cell with the ability to absorb things by wrapping its cell membrane around them. Another was a bacterial cell that converted solar energy into sugar molecules through photosynthesis. A third used oxygen gas to break down materials like sugar and release its energy into a form useful for life activities. The blob cells would occasionally absorb the little photosynthetic bacteria. These bacteria then lived inside the blob and divided like they always had, but their existence became linked. If you stumbled upon this living arrangement, you might just think that the whole thing was one organism, that the green photosynthetic bacteria were just a part of the blob that performed one of its life functions, just like your heart is a part of you that performs the function of pumping your blood. This process of cells living together is called endosymbiosis, one organism living inside another. But the endosymbiosis didn't stop there. What would happen if the other bacteria moved in too? Now the cells of this species started becoming highly complex. They were big and full of intricate structures that we call chloroplasts and mitochondria. These structures worked together to harness sunlight, make sugar, and break down that sugar using the oxygen that right around this time started to appear in the Earth's atmosphere. Organisms absorbing other organisms was one way species adapted to the changing environmental conditions of their surroundings. This little story highlights what biologists call the endosymbiotic theory, the current best explanation of how complex cells evolved. There's a lot of evidence that supports this theory, but let's look at three main pieces. First, the chloroplasts and mitochondria in our cells multiply the very same way as those ancient bacteria, which are still around, by the way. In fact, if you destroy these structures in a cell, no new ones will appear. The cell can't make them. They can only make more of themselves. Second piece of evidence. Chloroplasts and mitochondria both contain their own DNA and ribosomes. Their DNA has a circular structure that is strikingly similar to the DNA of the ancient bacteria, and it also contains many similar genes. The ribosomes, or protein assembly machines of chloroplasts and mitochondria, also have the same structure as ribosomes of ancient bacteria, but are different from the ribosomes hanging around the rest of the eukaryotic cell. Lastly, think about the membranes involved in the engulfing process. Chloroplasts and mitochondria both have two membranes surrounding them, an inner and outer membrane. Their inner membrane contains some particular lipids and proteins that are not present in the outer membrane. Why is that significant? Because their outer membrane used to belong to the blob cell. When they were engulfed in the endosymbiosis process, they got wrapped up in that membrane and kept their own as their inner one. Surely enough, those same lipids and proteins are found on the membranes of the ancient bacteria. Biologists now use this theory to explain the origin of the vast variety of eukaryotic organisms. Take the green algae that grow on the walls of swimming pools. A larger eukaryotic cell with spinning tail structures, or flagella, at some point absorbed algae like these to form what we now call euglena. Euglena can perform photosynthesis, break down sugar using oxygen, and swim around pond water. And as the theory would predict, the chloroplasts in these euglena have three membranes, since they had two before being engulfed. The absorbing process of endosymbiotic theory allowed organisms to combine powerful abilities to become better adapted to life on Earth. The results were species capable of much more than when they were separate organisms. And this was an evolutionary leap that led to the microorganisms, plants, and animals we observe on the planet today.
Okay, so make sure that you're comfortable with those three pieces of evidence of the endosymbiotic theory. Go ahead and stop your video now and make sure on page 11 you've taken those notes in the bottom part of the page and be ready to report back those three pieces of the endosymbiotic theory in class. Make sure that if you need to go back, you can rewind or go back in the video and make sure you get them. Hope that was helpful, guys. Bye.